It's nice to see you again, Udit.
Same here, Tina. How are you?
Very well. How are you?
Excellent, excellent.
Thanks for taking the time to talk a little bit about our favorite technologies. And I have some questions that I think we outlined ahead of time, but I'm sure maybe a couple additional ones will emerge as our conversation
You take it
goes
where
on.
you want to. I had a chance to look at those questions, and you can take it wherever you feel you're comfortable and whatever helps.
Great. The first question is one that always interests me based on my own experience with HPLC, and just to get um, your take on what you think the most recent important advances have been in the field, and both HPLC, but then of course the companion technology, mass spectrometry as well.
Yeah. Firstly, thank you again for having us, and thank you for giving us the opportunity to talk about technology with you. And I mean, it's always a pleasure. Look, I think you have to take a step back before you talk about technological advances and ask yourself, what are the big needs in the industry right now, right? And they're somehow paradoxical. So on the one hand, if you go into quality assurance, quality control, the home of HPLC now, there is still a significant need to simplify while maintaining robustness, right?
Yeah.
Right. So that's one extreme. On the other hand, you have the complexity of molecules. Roughly 45-50% of the pipeline now is novel modalities in biologics. The complexity of molecules warrants a multitude of different analytical techniques to be used to characterize them, right?
Yeah.
And so you have to walk this paradox when you're an analytical instrument company. On the one hand, you have to develop instruments that are simple, that somebody like me can use, and the data integrity is of the highest quality. On the other hand, you have to have any instrument that is required to analyze viral vectors and cell and gene therapy and oligonucleotides, whatever new modality that our colleagues in drug discovery are coming up with. So if you take these in turn, right, so take simplicity and robustness. I think in QA/QC, there's no doubt that operators are getting, we are able to attract people with less qualifications into these settings. What does that mean? Then that means that the instrument itself has to be sophisticated but simple to use, right? And Alliance IS, which is a product we've introduced, reduces errors in QA/QC by 40%. While maintaining the robustness that comes with data integrity that comes with using Empower, the software that is used to submit data to regulators, and reducing these commonplace errors reduces the burden on follow-up, reduces the burden on resubmission to regulators, as you well know, right? You've been in that industry, and it is pretty onerous to get even one error because you had the wrong solvent, or you had the wrong column, or you connected the wrong tube, and the instrument now prevents from happening. And a 40% reduction means that 6 out of 10 times it's actually a real error and real issue with the sample and not with the instrument. And that's fantastic, right? So it is the single most meaningful advance in over a decade in that space, right? And our customers are telling us that the adoption is pretty robust. Now, if I switch to the other, sorry, go ahead.
Yeah, before we go to the discovery and the sort of the other spectrum, the sophistication and new methods, new characterization, and do you see enough progress with on the regulatory and compendial adoption side? I mean, I think both of us remember what a journey the UPLC adoption was, and obviously, as you keep making progress in terms of automation and data integrity systems and all of that, the regulators and the compendia have to move with you for the users really to reap the benefit. Do you see that, or do you see that improving and accelerating, or is there still a way to go?
I think acceleration would be an overstatement, and one would not want acceleration in that space, right? I mean, nobody wants medicine to be approved on methods that are that are not necessarily robust and true and dried. But recently, the USP came up with a way of taking old HPLC methods and moving them onto UPLC. As you know, the speed of measurement increases dramatically if you do that. And that's a step in the right direction, right? We are in constant conversation in almost a tripartite way, right? With manufacturers, regulators, and instruments and systems manufacturers all together at places like Nimble, where we sort of sit in the same sort of space, where Waters has a lab at the University of Delaware, surrounded by folks from these different areas. And we have discussions on how can we take something like multi-angle light scattering, how can we take something like mass spec, simplify it enough so that it's measuring the CQAs that are necessary to approve the product while not complicating the workflow. How can we do that in such a way that it adds value? And those discussions are ongoing. I mean, it's a long process, as you know, but I'm optimistic. I think it is not for the lack of trying, and it is not for the lack of intent and collaboration across every domain, right? And the step from USP is a good one, right? It's a step in the right direction, and I mean, you save a lot of time. You have robust instruments, and now you save a lot of time in QC.
Absolutely. I agree. And it has to take the time that is needed to get the robustness and around those scientific discussions. But let's go back to the super sophisticated, the discoveries, the end, sort of the bleeding edge of discovery. What do you see there? Where do you see the needs, and where do you kind of see that next frontier?
I think, again, I mean, Waters is a company that spends most of our time in taking these highly complex techniques and simplifying them, right? And simplification, don't get me wrong, simplification is still required even if you're upstream. Even in proteomics, even in measurements for cell and gene therapy, you want a simpler technique. So let's take an example of empty versus full viral capsids, right? A key issue in cell and gene therapy, and a pretty important one, right? So you take a step back, you ask yourself, what are the biggest challenges in that area? Number one is cost, and number two is the regulatory burden, because this is a new entity. The regulators want a lot of information on it, right? So take cost for a minute. Today, roughly 30% of the sample is used for assay development in a single run. And these are sort of batches that are for n equal to 1 patient. So 30% goes to an assay development lab. And by the way, COGS are between $500,000 to $1 million. So this is a substantial amount of cost, right? How do you reduce it? I mean, you can reduce it by reducing the amount of sample. You can do that by decreasing the number of tests you do. Of course, you don't want that if you want if you have increased scrutiny on safety and different attributes, or you can reduce the volume per test, and the number of tests. And we can do so, for instance, with multi-angle light scattering, right? With multi-angle light scattering, you can take 4 assays like ddPCR, ELISA, mass spec, and maybe one other assay that slips my mind for now. You can take 4 assays and convert them into one and get the same information with a fraction of the sample without destroying it, an AUC, an analytical ultracentrifugation. And you can get important information about the sample, same information about the sample with a small amount of, same information about the drug with a small amount of sample, so saving time, while giving the researcher the same amount of confidence in the quality attributes they're measuring. So for instance, they're trying to look at aggregates. They're trying to look at empty versus full capsids. They're trying to look at denatured proteins, right? So you can get the same information with with one technique. And I think the idea is not to complicate the lives of people upstream. The idea is to simplify it. And as you and I have been in those labs as well, who wants to measure 15 different things and then aggregate the data and come up with cross correlations? If you have one piece of information that gives you a clear idea of an empty versus full versus the aggregate versus unaggregated molecules, I think that saves you a lot of time and energy. So upstream, with newer modalities, I think what we really need to do is pay attention to what's being developed, look at the problem that needs to be solved, And that is not just the province of the researchers, it's the province of instrument manufacturers, analysts, the people who do analysis downstream, because you can make up your own mind on how to solve the problem. I don't think people who are developing these molecules are inherently going to be able to come up with a clear idea of what instrument or what analytical technique is going to give them the answer. They are posing a problem. They're saying the cost is high and I want to be able to get these 5 attributes. How do I do it? It's our task as instrument manufacturers to say, okay, I can give you a simple solution or I can give you a complex solution. So long answer, but it's something that we're working on day in, day out, and it's near and dear to my heart.
Yeah, and I think you make a really good point in terms of how you basically present and translate solutions also from, I guess, one product to another. When you mentioned the multi-angle light scattering, that's a technique that is where the usefulness or is already been shown for molecules like heparin, and you can now not easily, but it's clear that it has a whole new applicability with the capsid-based delivery system. So that's very exciting, and it leads me to my next question. And you talked about that a little bit already. Sort of, how do you about the horizon scanning to understand what are the next challenges on the horizon for the therapeutics manufacturers, for our industry, so that you can kind of be right in the middle of that development and facilitate those key key steps. How do you horizon scan as the leader, and how do you facilitate the horizon scanning for your innovation?
Yeah, you talk to a lot of people, you read a lot, you pay attention to details, and eventually you form your own point of view after listening and learning. And that's critical. The last piece is critical, right? Because the customer is not going to pose the problem to you in the way you need to solve it. They have a problem, the patient has a problem, the patient has a certain disease, which we're developing a therapeutic for. It's available in limited quantities. How the heck do we get it to the patient, right? So that's one type of problem. And you've got to talk to a lot of people, go to a lot of conferences, and I think eventually form your own point of view. And I can give you an example, perhaps several examples, right? I mean, take the development of something like MABs, right? I mean, we've known this for ages. These are naturally, with cell culture, these are naturally made molecules, right? I mean, you're using cell culture media, which is not usually naturally derived. So the ingredient, the raw materials are naturally derived. Then you're using different types of cells to generate the monoclonal antibody in cell culture. Then you're doing downstream processing. But inherently, there is variability introduced in step 0 in how you source the raw materials, right? So when there is variability downstream, you have to do the problem solving. You start downstream, but the problem could have occurred way upstream. So what customers are now doing with us is saying, hey, can I get a fingerprint of the cell culture media that I'm buying? And can I assess lot-to-lot variability? And I remember a time when, this was way back, probably 6-8 years ago, when one of our customers suddenly had the titer of their monoclonal antibody increase 30-fold from one batch to the next, right? And the head of, this was a few days into my job at MilliporeSigma, right? And I got a call from the head of manufacturing of one of these large pharmaceutical manufacturers who I knew very well. And he said, hey, Udit, this titer has increased 30-fold. I said, congrats, that's wonderful. You must have done something to the process. He said, no, this is out of spec. I won't be able to supply, and my CEO is on my back because we have inventory for 6 months. I got to fix this as fast as possible. And we think it's because of one of the raw materials you guys supplied us. I said, okay, I didn't realize that increase in productivity was a problem, but let's go figuring this out. It took us 3 months to figure out what the issue was. It was parts per million difference in a chelating agent, zinc, in the cell culture media, right? Yeah. Now, fast forward 6-7 years now at Waters, we're helping customers to fingerprint cell culture media using mass spec. right? And time of flight mass spec, and saying, okay, let's fingerprint this. If there's any variability, we'll do assessments upfront, right? And go ahead and use this in the process. But if you discover anything untoward, we would have already figured out what the issue is. It's a pretty blunt instrument, mass spec is, if you use it in that way, but it is exquisite in its ability to break down molecules and using algorithms to reconstruct what we had, right? So that's just one example of paying attention to what the challenge is and using your own brain to say, well, the problem is actually not downstream. The problem is in the raw material, because that is the most variable component in this whole stream. The cells are well characterized. The filtration is well characterized. The chromatography is well characterized. The viral filtration is well characterized. But we've got these cells and we've got cell culture media, right? And so that's just one example of being able to detect something and assess something way upstream to reduce problems downstream. And AstraZeneca has adopted that method, Johnson & Johnson has adopted that method, and there's a few others who are in line. As one does, others do as well. It's not a regulatory problem, it's a problem-solving approach.
That it is not a regulatory problem, but again, I think it's another area that I think the regulator still has to be aware and understand, that where the expectation as to what's happening, because you're managing a platform technology at that point, right? When you're making a monoclonal antibody and understanding the variables of that platform as a whole becomes a question for the regulator.